today's module, we're going to be discussing Mendelian or classical genetics. So we're going to be looking at how traits are inherited and predicting outcomes once these traits have been identified. So to begin with, I want to discuss some terminology that's important to these next couple of modules, and then we'll get into how these concepts were derived. So first of all, just reviewing a little bit, we've already talked about mitosis and meiosis, and so there is still an implication here with these topics as we discuss Mendelian genetics. So the basic concept, of course, is that our cells have two copies of chromosomes. We have diploid cells. Through meiosis, our gametes will carry one set of our 23 chromosomes, and then after fertilization, that reunites the diploid condition. So we go from diploid to haploid in the gametes, and then reestablish the diploid condition. So again, bottom line is that we inherit two sets of chromosomes. We get to pass on one set of chromosomes. So that's something to keep in the back of your mind as we look at inheritance. All right, again, looking at some terminology, we've talked about chromosomes. So hopefully we're comfortable with the concept of chromosomes and knowing that the DNA double helix is a genetic component of chromosomes. A gene is a portion of a chromosome that codes for a trait, a physical trait or chemical trait. And so, for example, we could be looking at plant chromosomes. These are P chromosomes. And if we were to look at a homologous pair of chromosomes, because again, there are two sets of chromosomes that are inherited. So for each chromosome type, there are two of each type, one coming from the male, one coming from the female. So if we were to look at these homologous chromosomes, then at a particular place, we could identify a particular gene. So in this case, there's a gene for flower color. And a gene will be found at a particular locus. So the gene for flower color isn't just scattered anywhere. I'll find it at a particular place on a particular homologous chromosome, and I would find it in exactly the same place on the other homologous chromosome. So chromosomes tend to match up, homologous chromosomes match up gene for gene. Now, another term I'm going to be using a lot is the term allele. So the term allele also means a gene. So again, a section of a chromosome coding for a particular protein or particular trait. But there's an implication when the term allele is used, and it is that there's more than one version of that particular gene. So again, looking at this example, we have a gene for flower color, but that gene comes in two alleles. One allele codes for purple flowers, and one allele codes for white flowers. So there are two possible alleles that we can inherit, or at least the plant that can inherit at that particular locus on that particular chromosome. And again, I'll use a convention here of what's known as dominant and recessive. So dominant alleles I'll normally represent with capital letters. So-called recessive alleles I will represent with lowercase letters. So in this case, purple is dominant over white, and we're going to find out more about what dominant and recessive means a little bit later. So again, P will be the dominant allele here. The capital P will be dominant, lowercase p will be recessive. And the thing I want to mention about this is that dominant doesn't mean better. Dominant is not stronger or better or anything like that. The difference between a dominant and recessive allele is that if the dominant allele is present, you will see it physically expressed. You will see evidence of it. Recessive alleles can be hidden. And so that's a very important concept to keep in mind. Dominant alleles always show themselves. Recessive alleles can be hidden. Now, alleles or genes then, we normally discuss in pairs because, again, chromosomes are inherited in pairs, and so for each chromosome you will have two alleles at a particular locus for a particular trait. So again, we're going to continue on with this theme of flower color. So as we look at the possible gene or allelic pairs then, it is possible that one could have two dominant alleles together. So the alleles could be the same. You could have two dominant alleles, or you could have two recessive alleles. And if that's the case, we describe that as a homozygous condition. Okay? So again, it isn't helpful to say PP, because if you say PP, I don't know if you're talking about dominant or recessive alleles here. So again, if they're both the same type of allele, either both dominant or both recessive, we would describe that as a homozygous allelic pair or gene pair. Now, if one is homozygous and one is recessive, we describe that condition as heterozygous. So again, the prefix Homo means the same. The prefix hetero means different. So same seed or different seed is essentially what the word is trying to tell us. Now, additionally, as we talk about these gene pairs, if both of the alleles are dominant alleles, they're not only homozygous, but we would describe that gene pair as a homozygous dominant pair. So again, PP isn't going to communicate much, but homozygous dominant tells me that both of the alleles are the same and they're both dominant alleles. Again, heterozygous we've already discussed. That would be one dominant and one recessive allele that has been inherited. And if you've inherited two recessive alleles, again, you would be homozygous recessive. So again, when you're trying to communicate these, and as I communicate these with you, I won't be using the letters as I describe them. I'll be using these three descriptions: homozygous dominant, heterozygous, or homozygous recessive to describe the gene pairs. All right, now, we, for the most part, will be talking about what's known as complete dominance. And also there are a couple other terms here that are related to complete dominance. And those terms are genotype and phenotype. So again, as we look at our three gene pairs, we can have homozygous dominant, heterozygous, or homozygous recessive. And that would describe the genotype. And genotype, essentially, that word is saying the type of genes that we have, or what type of gene pair do we have. Now, the genotype results in some physical appearance in the organism, and that's known as the phenotype. What is the physical expression of the genes? What type of physical appearance do you have in phenotype? So in this case, when we're looking at this flower color gene in the pea plant, again, we have three gene pairs, three genotypes, but we really just have two possible phenotypes with what's known as complete dominance. So if you're homozygous dominant or heterozygous, this is a plant that has purple flowers. And so by looking at the flower, you can't tell whether it's homozygous dominant or heterozygous. And so again, that's what makes a dominant allele a dominant allele. It expresses itself if it is present. And notice here that the recessive allele in the heterozygote is present, but it's being hidden. So again, you're just seeing purple flowers. You don't know what the genotype is. If you're homozygous recessive, that would express as white flowers. So homozygous recessive would be the genotype, and the phenotype would be white flowers. And notice that the only way you can show the recessive phenotype is to be homozygous recessive. And this is an important concept to keep in mind when we start solving problems, because you always want to start with what you know. If flowers are purple, you don't know if they're homozygous dominant or heterozygous. But if flowers are white, they are homozygous recessive, because that's the only way the recessive trait can show itself, is to be two recessive alleles. Okay. Now, the person responsible for figuring this all out was Gregor Mendel. Gregor Mendel is here, pictured here. And Mendel was a German monk who worked primarily in Czechoslovakia, did most of his work there. He initially started working with animals, but switched to plants a little bit later on, basically started a garden in the monastery, and was interested in pursuing inheritance. He was interested in pursuing this question. His superiors didn't really like him doing that, and so he basically started a garden to continue his investigation of these ideas. And so the pea plant essentially was his tool, his research tool. And so what he would do is control the breeding of his plants by cutting off the male parts of one flower and leaving the pistil or the female organ there. And then he would take pollen from the male organs, the stamens of one flower, and then he would brush those onto the female part or the pistil of another flower and control breeding. So it wasn't random breeding, he was controlling the breeding, he was preventing self-fertilization of flowers and then deciding who breeds to who. So it was a very meticulous process, as you can imagine. But he was a meticulous guy, so that's the way he was. So what he was interested in was looking at what was known at that time, the primary theory of inheritance was the blending hypothesis, which was basically that if you have two extremes and you breed them together, you'll get some intermediate form. And so again, Mendel was an interesting guy. He came from a common background, but he had a pretty good education, had gone into the clergy, and had a very good mathematical background. He had a gift for mathematics. He also had a real gift for nature as well, and a keen eye for detail. So as he looked at his pea plants, he began to notice either-or conditions in his pea plants. And so he wanted to test this blending hypothesis when he saw these either-or conditions. So one of these either-or conditions he identified were flower color. So he looked and observed either purple flowers or white flowers in the pea plants that he was gardening. And so what he initially did then was establish a garden where whenever he bred purple flowers with purple flowers, he always got purple flowers. And so this is what's known as a P generation, the parental generation, and we would call this a true breeding generation as well. And what he didn't really know at that time, but later figured out, is that this was a homozygous dominant population, and I'll come back to that later. And then he established another population where there were white-flowered plants, and anytime he bred the white-flowered plants, he always got white-flowered plants. So this was a separate population, again, that was also homozygous, in this case homozygous recessive, although he again figured this stuff out later. So then he decided to go ahead and breed members of his pure breeding purple population with his pure breeding white population. Now, if it's the blending hypothesis, you would expect some kind of a light purple, type flower. And what he got was purple flowers. Excuse me. So the entire offspring, the so-called F1 generation, all of the offspring, if he crossed the purple and the white flowers, were purple. So right away, that disproved the blending hypothesis. So he was, curious as to what was going on here. So then he took these F1 plants and allowed them to pollinate with one another, or he pollinated them with one another, crossed them with one another. And what happened there is that the white trait reappeared. So in the parental generation, we had white flowers. They disappeared in what's known as the F1 generation, the first filial generation. The white trait disappeared. And then in the so-called F2, or second filial generation, the white trait reappeared. So what he determined was that this trait could be hidden and then reappear in the next generation. And he also then noticed that the purple to white in the F2 generation was a 3-to-1 ratio. And again, being a mathematician, he began to look at these ratios and began to think about what would be necessary for some physical material going to generation to actually generate going from having traits to disappearing traits and reappearing in a 3-to-1 ratio. So what he ultimately figured out from that was that traits come in twos, and that you could pass on one version of your gene, and then that would be combined with one from the other parent to reestablish the diploid condition in the next generation. So he referred to this as segregation, that you have two copies of genes, you get to pass on one copy. And then your partner passes on one copy, and that gets you back to two. So again, he knew nothing of mitosis, meiosis, DNA, chromosomes, any of this stuff. He basically inferred all of this from looking at the ratios. And also, he came up with the idea then that one type of allele would be dominant over the other so that alleles could be hidden, that traits could be hidden in this way. So again, he inferred all of this mathematically, which is, his genius and why he is considered a genius. Now, he then began to look at other traits. So he initially started this with, and we're talking about flower color, but he identified a number of different traits that came in these either-or conditions. And he tested all of these in the same way. He established pure breeding populations, he crossed them, and then kept seeing the same results over and over again. So again, he worked with flower color, purple versus white. He also looked at flower position. Flower positions were flowers, so-called axial flowers that were produced at the nodes, as opposed to terminal flowers, which are produced at the tips, so axial dominant over terminal. And again, what he found was that the terminal flowers disappeared in the F1 and then reappeared in the F2 in approximately a 3-to-1 ratio, and so the actual numbers are here. Yellow versus green seed color, again, same idea. When he crossed these, green seeds disappear in the F1, they reappear in the F2 in a 3-to-1 ratio. Smooth seeds or round seeds versus wrinkled seeds. Again, wrinkled seeds disappear in the F1, reappear in the F2 in a 3-to-1 ratio. Also looked at inflated versus constricted pods, green versus yellow pods, and then also size. Plants are either very tall, 6 or 7 feet in length, or less than 2 feet in length. And so again, regardless of which of these traits he was looking at, one would disappear, then reappear in the F2 in a 3-to-1 ratio. So again, he kept seeing these ratios over and over again, and this eventually led him to the concepts of dominant versus recessive, and the idea that you have two sets of genes, pass on one, and those are matched up with the mates to reestablish the two. So pretty amazing given that he didn't know anything about molecular biology at all. So again, the traits he looked at: flower color, seed shape, seed colors, pod shapes, and stem lengths. These were all characteristics where he got exactly the same results all the time. And again, he did not see blending in any of these. And the reason for that is that we were looking at this type of inheritance known as complete dominance, where there's no difference between the homozygous dominant and the heterozygous. Okay, so let's go ahead then and look at the basic traits. There are basically six basic crosses rather. So let's look at the 6 basic crosses that we see in genetics. And basically, all of the things we're going to come back to come down to these 6 basic crosses. So what we're interested in here is what are the genotypic ratios generated in these crosses and what are the phenotypic ratios generated in these crosses. So let's start with homozygous dominant times homozygous dominant. The way we do these problems is set up what's known as a Punnett square. You'll set up 2 axes. And what you want to list out on each axis is what you would expect to find in the gamete for this particular gene of a particular chromosome. So in other words, you have 2 genes. You can only pass on 1. And so half of your gametes will contain this allele. Half of your gametes would contain this allele. So all of the gametes in this case would have a dominant allele because that's all there is. That's all you have to pass on or all this individual has to pass on. The other individual would make gametes that have this allele or this allele, and of course they're both dominant alleles, so all of the gametes there, 100% of the gametes there, would contain dominant alleles. So if 100% of the sperm have dominant alleles and 100% of the eggs have dominant alleles, and these came together randomly, there's only one possible outcome, which is homozygous dominant. So when you cross homozygous dominant times homozygous dominant, there's only one genotypic outcome, 100% homozygous dominant. And phenotypically then, all of your population offspring would be purple. I don't think that's a big surprise. So this is really what the pure breeding population that Mendel had established, the purple plants, this is what was going on there. They were just passing around dominant alleles, so there was no possible outcome other than homozygous dominant in the offspring. Now you can also cross homozygous recessive with homozygous recessive. And again, if you set up a Punnett square, you could package gametes with this or this allele. They happen to be the same allele, so we'll just list it once. The mate can package gametes with this allele or this allele. So again, 100% of the gametes would carry the recessive allele here, and 100% of the gametes will carry the recessive allele here. So if any of these gametes fuse with any of these gametes, so any of these sperm fuse with any of these eggs, the only possible outcome here would be homozygous recessive. All of the offspring will be homozygous recessive, and 100% of the offspring then would have white flowers in this situation. And again, that's what was going on with the pure breeding white population. So they were being crossed repeatedly. Next, Darwin decided to cross a member of his pure breeding homozygous dominant population with a homozygous recessive population. And if you do that, again, you would have half your gametes carrying a dominant allele, half your gametes carrying a dominant allele, so 100% of your gametes from this individual will carry a dominant allele. This individual, half the gametes will carry a recessive allele, the other half of the gametes will carry this recessive allele, so 100% of the gametes carry a recessive allele. So if any of these sperm fuse with any of these eggs, again, only one possible outcome, which would be heterozygous. And phenotypically, 100% of the offspring would be purple. So the white trait disappears then in that generation, and again, that's what Mendel found. When he crossed homozygous dominant with homozygous recessive, you got 100% purple. They're all carrying the recessive allele, but they're not showing it because it's hidden by the dominant allele. So again, dominant alleles show themselves, recessive alleles can be hidden. That's what defines a recessive allele. Okay, so then going into the F2, what Mendel was doing then was crossing heterozygotes. So let's see what happens when we cross heterozygotes. What would be the possible outcomes there? So again, we'll set up our Punnett square. So now we have some differences. So here, again, you have 2 alleles. You can only put one into a sperm or an egg. So in this case, half the alleles will carry, half the gametes will carry the dominant allele. The other half of the gametes would carry the recessive allele here. So half will carry the dominant allele, half will carry the recessive allele. And the same with this parent. Half the gametes will carry the dominant allele, half the gametes will carry the recessive allele. And again, we can't control what sperm and eggs come together, so that's a random process, and that's what's important to know about these laws of probability, is that they're random laws of probability. So it would be possible for this sperm and this egg to fuse together, which would give us a homozygous dominant offspring. It is possible for this sperm and this egg to fuse together, which would give us a heterozygous outcome. It's possible for this sperm and this egg to fuse together, giving us heterozygous once again. And it's possible for this sperm and this egg to fuse together to produce homozygous recessive. So as we look at the outcomes here then, we would expect 1 out of 4 of the offspring to be homozygous dominant. We would expect half of the offspring to be heterozygous, because we have 2 heterozygous outcomes out of 4 possibilities here. And we would expect 1/4 of them also to be homozygous recessive. So that would be the genotypic ratio or expected outcome here. And then phenotypically, we would expect 3/4 to be purple and 1/4 to be white. So what's important to understand then is that these are probabilities. When you have lots of outcomes, you tend to see that. So for example, if you flip a coin 2 times, are you guaranteed of getting 2 heads? Well, no, you're not, of course not. So that's the difference between probability and reality. So these are probabilities. These are expected outcomes. 1/4 homozygous dominant, half heterozygous, 1/4 homozygous recessive, 3/4 purple, 1/4 white. But again, if you flipped a coin 1,000 times, you are likely to see about a 50-50 distribution in that. So the more you do it, the more likely you are to see the expected outcomes. But there's no guarantee that you see the expected outcomes, especially when you have only a few outcomes to work with. So again, if you cross heterozygotes and there's only 1 offspring, what would it be? Well, the best chance would be that it would be heterozygous and show a purple flower in this case. But you're not guaranteed that outcome. So that's the difference between probability, prediction, and reality. They don't always match up. But fortunately for Mendel, when he did his experiments, they did match up, and that helped him figure out what was going on. Okay, so again, in the F2 then, what Mendel was seeing was homozygous dominance and heterozygous, all of which had the same phenotype in a 3-to-1 ratio compared to homozygous recessives. Okay, now, we still have a couple other possible crosses here, and these aren't ones that Mendel necessarily did in his experiments, but there are some other possible crosses. So one of these would be if you cross a homozygous dominant with a heterozygous. So again, we would set up our Punnett square here. Okay, this parent here could be, half of the gametes would carry a dominant allele, the other half of the gametes would carry the dominant allele, but we only list it once. We don't need to list it twice when 100% of the gametes are the same. And then this individual here, half the gametes could carry a dominant allele, half the gametes could carry a recessive allele. So we would list those both separately because they're different. And again, this sperm could fuse with this egg, this sperm could fuse with that egg, and that would be a random possibility. So we would expect half of our offspring to be homozygous dominant, half of our offspring to be heterozygous, and all of the offspring to be purple in this particular case. Now, still one other cross would be if we cross homozygous recessive with heterozygous. And again, if we set up our Punnett square, half the gametes would carry a recessive allele, the other half of the gametes would carry the other recessive allele, so 100% of the gametes would be carrying a recessive allele. And here, half the gametes would be carrying a dominant allele, half the gametes would be carrying a recessive allele. And so, again, this sperm could fuse with that egg, this sperm could fuse with that egg. Equal likelihood that either could occur, so we would expect, again, half to be heterozygous, half to be homozygous recessive, meaning half would be purple phenotypically and half would be white phenotypically. So, again, as we look at the possible crosses then, basically all genetic problems are going to come down to these 6 basic crosses. And so ideally you memorize the genotypic ratios and the phenotypic ratios, but if you should forget them, you can always set up a simple Punnett square and figure them out. So it's very important that you put pencil to paper with this, pen to paper with these and figure these out. So again, our 6 crosses would be homozygous dominant times homozygous dominant, homozygous recessive times homozygous recessive, homozygous dominant times homozygous recessive, heterozygote times heterozygote, heterozygote times homozygous dominant, and heterozygote times homozygous recessive. So again, all crosses will come down to those, or essentially all genetics problems we can bring back to these 6 basic crosses. Now, I want to come back to dominance real quickly. Again, we're talking about what's known as complete dominance. And so assume a complete dominance unless I tell you otherwise. But with complete dominance, we're talking about a single gene or a single pair of alleles then, we cannot tell the difference between homozygous dominant and heterozygous. They're going to look the same. So the phenotype doesn't tell us the genotype. It gives us a clue as to genotype, but it doesn't guarantee the genotype. Whereas, again, if they show the recessive phenotype, it does tell us the genotype. So the only way then to show recessive phenotypes is to be homozygous recessive. So again, this is a foundational concept when we begin to solve problems. Look for the recessive phenotype. Whoever shows it is telling you their genotype. And then we may be able to figure out other genotypes based on that, depending on what alleles have been inherited and what alleles are passed on. But again, we can't tell the difference between homozygous dominance and heterozygous. Now, what's interesting is that there is another type, and so again, we have two distinct phenotypes, dominant phenotypes and recessive phenotypes. Now, if you have then an organism that shows the dominant phenotype, and if you want to determine whether it's homozygous, dominant, or heterozygous, then what you can do is what's known as a test cross. And so what a test cross is that you will take your dominant phenotype, in this case plant, and you'll cross it with a homozygous recessive. And again, how would you know if it's a homozygous recessive? Well, it will show the recessive phenotype. The only way you can show the recessive phenotype is to be homozygous recessive. And if we go back to our basic crosses, then if you crossed a homozygous dominant with a homozygous recessive, then all of the offspring would show the dominant phenotype. In this case, they would all be purple. And if this individual was a heterozygote then, and you crossed it with a homozygous recessive, we would have half purple, half white. So again, this goes back to our basic Punnett squares. You may want to review that if that isn't obvious to you. But again, the test cross, what happens if you cross with a homozygous recessive, if you see the recessives in the next generation, then that's telling you that this parental type was heterozygous. If you don't see them in the next generation, it's telling you that this parental type was homozygous dominant. So that's what we call a test cross. So getting back to Mendel, and Mendel was a pretty amazing guy in a lot of ways. Again, he knew nothing of molecular biology, chromosomes, genes, any of this, yet he figured out basically the difference between haploid and diploid and what meiosis is all about, that we inherit two sets of chromosomes, we pass on one set of chromosomes, meaning that we inherit two alleles and get to pass on one allele. And then our partner passes on another allele, that gets us back to the diploid condition in the next generation. So unfortunately, Mendel was not appreciated in his own time, but his work was rediscovered early in the 20th century, and he ultimately became famous after his death.